I'm going to let you explore our topic today before I bother talking about it. So what we're talking about today is glycolysis. And so we have this very short, easy to recognize, I think, clicks. It's one of our CK12 activities. So in Google Classroom, you're going to find products of glycolysis. It is the one that is, there's a lot that I posted today. They're all small items. It's the one that's due at noon. So open that one up. We're going to be in groups. I'm going to open up the breakout rooms so that you can kind of discuss it as you go. I'd of course like somebody to share their screen, and then you all can go through the process, but I want you to talk through the questions and when you do the animation, the manipulative, it tells you to like grab the red dot and drag it. I want you to talk about what do you see happening, and then answer the questions together. So you can talk about those questions. And if I could get this bottom half of my screen to turn their screens on, remember when we're in breakouts, it is polite to turn on your screens and your sounds. As a matter of fact, at the beginning of the year, when we talked about the different norms, if you will, of virtual learning, cameras was one of the things that the majority of you identified as being important. So it's, as the year gone on, some of you have kind of lost track of that norm. So maybe we bring that back together, because Sophia doesn't want to be in the room by herself, right? She wants to be with people who will talk to her and who she can see. So you'll open up that Plix. I'm going to open up breakout rooms. And remember, if you updated your Zoom, you should have the option to. So I'm going to, you know, when we're in real class, I ask questions and you guys answer questions, and we do it just kind of out loud, and I don't look for it in a chat, and I don't have to elicit it through an earpod. So I'm just going to ask you some questions and you guys voice up and answer like you're in a class, okay? So in this activity, you started with glucose and you went through a process of chemical reactions. And I'm wanting to know what happened to glucose during this activity. Sartaj, what happened to glucose during this activity?
Motivated state, it was split. So it started with 6 carbons and it ended with 3.
That's perfect. Yes. So we have glucose, and each of these white dots were carbons. And in the process, we see the carbon is split. Glucose is split, and in the end, you have 3 carbon compounds. This molecule is called pyruvate. So come test day, we want to know what happens to glucose during glycolysis. It's split in half and it forms 2 pyruvates. Perfect. Which has more energy, do you think? These 2 pyruvate molecules or this 1 glucose molecule? What are your thoughts on that? Which has more energy? 1 molecule of glucose or 2 molecules of pyruvate? Taylor, what do you think?
The 1 molecule, because it gets split into 2.
Okay, and we've learned over and over again, we lose energy when we break bonds. Perfect. So 1 molecule has more energy than these 2 3-carbon molecules. I feel like that was a test question last year. So in this process, is ATP used or is ATP produced? What did you notice? Was ATP used or was ATP produced? What do you think, Elise?
It was produced.
It was produced, but it was also used. So it's kind of a trick question there. So what I see, the orange balls, right? Those are my ATP molecules. Notice in the very first step, those 2 ATP molecules are being used. So this actually is like there are a number of reactions. This is showing you as it progresses, okay? So 2 ATP are initially used. And as I go, those ATP are gone. Now watch. Now 2 ATP have come out. And at this point, 2 more are used or are come out. So in the end, 4 ATP molecules come out. 2 went in. You made 4. So you say you have a net gain of 2, right? 4 minus 2. We get 4, but we use 2. So really, we only get 2 out of it. Okay. This green molecule, what does that remind you of? NAD+. Does it remind you of a molecule we used last section? NAD+. What is that similar to? What do you think, Sophia? Just naming wise, we had another one that was similar.
NADPH?
Yeah, correct. So we had NADPH and now you have NADH. So this gets confused a lot because they're so similar. P, keep it with photosynthesis, right? Photosynthesis starts with a P, so NADP stays with photosynthesis. They serve the same purpose. They're both electron carriers. So notice we have NAD going in, okay? And as we proceed, 2 ATP are used, 2 NAD pluses are used, and when they come out, they must have picked up a hydrogen and electrons, and now they're NADHs. So how many NADHs did we make in the end? Ellie?
2.
Yes. So basically what you want to know about glycolysis is what goes in and what comes out, okay? You guys are going to make all of that wonderful, oh wait, I'm recording, good, okay. So you are going to make a manipulative today, showing me your understanding of glycolysis. So you're going to show me how NAD turns into NADH and how ATP is used and glucose turns into pyruvate, okay? So we're going to walk through those steps a little bit together. You have a student version if you'd like to use it. Anyways, I don't make copies of those student versions for everybody because not everybody wants them. But all you have to do is click, open it and then select File, make a copy. Now you have your very own copy that you can edit, okay? So if you want to use the student versions, go ahead and make a copy of that. In the end, we'll do a Kahoot. You can choose to stay or you can choose to get started on your homework, which will be reading out of the CK12. That's due today. And then your manipulation of glycolysis will be a video that you turn in Flipgrid by tomorrow, okay? So any questions about the overall agenda? Very good. So let's get into cellular respiration. A number of steps are involved in cellular respiration. We're going to break them up. Today, we're going to talk about only the first part, which is glycolysis, which you saw in that Plix. Pretty easy, right? Not a lot's happening, but we're going to break it down step by step. So we'll talk about glycolysis today. We turn glucose into pyruvate, which is also called pyruvic acid. So different resources will use the different version of the name. Our next lecture, we'll talk about the Krebs cycle and the electron transport chain where we make the majority of our energy. So today, just this yellow part, just the glucose or glycolysis, which literally means sugar splitting, breaking apart, lysis. Notice we have a memorize this formula. You had that on your photosynthesis test, right? We said memorize this, and then it showed up and it was worth 2 points. Basically the exact same formula, only backwards, right? So the products of one are the reactant of the other. So we start with glucose plus 6O2, which we won't use today. We'll use it in the next lecture. Yields 6 CO2 plus 6 waters plus ATP energy. Okay, so memorize that, highlight it, circle it, asterisk it. It's a guaranteed test question. You will need to know the 3 processes that are involved in cellular respiration. Circle that. That's guaranteed to be asked on a test. Basically, overall, you're going to break down glucose and make energy. And we know the energy that our cells use comes in the form of ATP. All right. So here are those different steps we talked about. This is the mitochondria. Do you remember? The mighty mitochondria makes all of our energy. Okay. So the process we're talking about today happens outside of the mitochondria. It's happening out in the cytoplasm. Glycolysis doesn't require any energy. I'm sorry. Glycolysis doesn't require any oxygen, and it's out in the cytoplasm. The portion that requires oxygen happens inside the mitochondria. Okay, so that's another day. So when we talked about, oops, we got another person waiting to come in. When we talked about thermodynamics, we identified that it's the study of the changes of energy within a system, right? So the change of energy in this case is glucose. And for every gram of glucose, we can release 3,811 calories. I bring this up because in chapter 6, we talked about carbohydrates releasing 4 kilocalories per gram. So this, you can see, is kind of a rounded off version of 3,811. That used to be a test question. I don't know that it will be this year. But what a calorie is, and I think this is repetition. I feel like we've said it before. It's the unit of heat energy required to raise 1 gram of water, 1 degree Celsius. So when they're talking about calories, they're really talking about heat energy. And that's the literal definition. 1 gram of water, 1 degree Celsius. Taylor remembered that energy was stored in bonds and released when bonds are broken. So this is, again, a reiteration that we've continued to use all year. I want my toolbar not to take up my whole screen. So this is glycolysis. What you guys saw in your animation, you saw the beginning and a generic version of the middle, and then the end, okay? So we're gonna look and see what's actually happening in those different places. Okay, so know that it happens in the cytoplasm. This is a guaranteed test question. Circle it, asterisk it, highlight it. Basically, you're starting with glucose and you're ending with pyruvate. So this first part is an investment phase. We're going to invest 2 ATP energies in order to get 4 out, okay? So glucose plus 2 ATP is going to yield these 2 3-carbon compounds. Do you remember PGAL from the last chapter? Right? So it makes sense if glucose or glycolysis is opposite photosynthesis, it would make sense that if I use PGAL to make glucose, I'm going to get PGAL when I break apart glucose, right? So I'm not yet to pyruvate. The ATP donates a phosphate on each end, and that's what you see here. And that's what gives it the ability to break that bond. So the energy used breaks the bond in that 6-carbon compound. So like in class, we usually do a manipulative and we have M&Ms and we have like 6 M&Ms, then we add ATP and then we break our 6 in half. So that's what's happening in that investment phase, that first step. In the next step, this is our payoff. We're going to get 4 ATP molecules. We use 2, so it's a net gain of 2. So the way we get those ATP molecules, here, the first piece, NAD+, is going to strip off a hydrogen and an electron from PGAL. That opens up a bond where this inorganic phosphate can be added. So the inorganic phosphate now you can see is here. So this is happening to both halves. NADH strips off that hydrogen, leaving open a bond for phosphate to bind. In the last step, we take off each of these phosphates will become an ATP molecule. Each phosphate comes off, becomes an ATP molecule. 1, 2, 3, 4 phosphates become 4 ATPs. Without the phosphates, this molecule is now pyruvic acid or pyruvate. Okay, so what goes in, what comes out? Glucose goes in, 2 ATP and 2 NAD+. What comes out? 2 NADH, 2 pyruvates, and 2 ATP. Okay, that's our process. So these phosphates are not tied up with ADP. They're inorganic. They're free-floating. They get recycled. NADH is formed along the way, as well as our pyruvates and ATP. We are assigned the task today of creating our own visual. So this is a process, and I think walking through the process is very helpful. So you create your own animation, manipulation, however you want. Use Cheerios and M&Ms and whatever you have around, screws, coins, Legos, anything, okay? So you're gonna make your own animation of that, and you're gonna make a video explaining it, and you're gonna put that into Flipgrid. So glucose goes in, 2 ATP go in, 2 NAD+ go in, 2 pyruvate, 4 ATP, and 2 NADH come out. So that's glycolysis. That's all we're gonna talk about today. We'll hit the rest of this starting tomorrow, okay? So what questions do you have on that?